Hey everyone! Today, I'm diving deep into the fascinating world of molecular biology to explore how prokaryotes, those tiny powerhouses without a nucleus, replicate their DNA. Now, if you're a biology student, you might already know that prokaryotes, like bacteria, have a much simpler structure compared to eukaryotes, which include humans and plants. But when it comes to DNA replication, things are still pretty complex. So let's start at the beginning. Prokaryotic DNA replication is bidirectional and originates at a single point in their circular DNA molecule called the origin of replication, or ORAC. Enzymes play a huge role here, especially an enzyme called DNA helicase. This enzyme unwinds the DNA, creating what we call a replication fork. At this fork, a bunch of other enzymes come into play. One of the key players is DNA polymerase, which adds nucleotides to build new strands of DNA. But it's not as simple as just adding pieces to a puzzle. These enzymes need a primer to start, usually a short RNA sequence made by another enzyme called primase. Now, because the strands of DNA are anti-parallel, replication happens a bit differently on each strand. The leading strand is synthesized continuously, while the lagging strand is synthesized in short bursts, creating what are known as Okazaki fragments. What's truly fascinating is how these Okazaki fragments are later joined together. This task is handled by DNA ligase, which seals the fragments into a continuous strand. It's like watching a molecular construction site where everything has to fit perfectly. And here's something to blow your mind. All this happens at a blazing speed. Prokaryotes can replicate their entire genome in just 40 minutes. Imagine copying all the information needed to build and run a living organism in less than an hour. To ensure everything goes right, prokaryotes have various proofreading mechanisms in place. These mechanisms correct errors during replication, which is crucial because errors can lead to mutations, and in a worst-case scenario, harmful ones. In summary, DNA replication in prokaryotes is a finely tuned process involving multiple enzymes and stages. It's a brilliant example of biological efficiency and precision, showing us just how incredible the mechanisms of life are. That's it for today's deep dive into the molecular biology of prokaryote DNA replication. If you found this fascinating, don't forget to like, share, and subscribe for more awesome content. Until next time, keep questioning and keep learning.